So in this course, we're going to be talking about some of the tools that are available and methods for analyzing large-scale genetic data, in particular with a focus on SNP data as well as sequence data. So we're going to aim to cover some of the basics of case control association testing, where you have a trait that is dichotomous, that you're interested in determining whether or not particular variants or variants of interest are associated with your output, which is the disease or no disease. We're also going to talk about genetic association testing with quantitative traits. Also, as far as like some of the methods available for gene and pathway level analysis. We're also going to talk about how to use genetic data to infer ancestry, population structure, and ancestry differences among individuals, which is often an important issue in genetic association studies where there can be confounding due to ancestry differences among individuals. So we're going to talk about how to infer ancestry as well as how to adjust for it when you're doing genetic association testing and some of the methods that are available out there for that. We're going to also cover rare variant analysis and the collapsing of tests. If you haven't been doing any sequence studies, then this is like some of the main ways to analyze sequence data. Also we'll talk about supervised burden tests and variance controlling tests for rare variants, omnibus tests, weight and design consideration. And then we're going to end with some material on gene-environment interactions, meta-analysis, and some of the emerging issues with high throughput sequencing data. So most importantly, as I said in the introduction, this is the class website. So this is where you're going to find the PDF copies of all of the slides. So the slides that you were given are not in color, but the PDF slides that are on the website are in color. So that's provided for you. So data sets that we're going to be using for some of the labs are also going to be available here. For some of the big data sets that may take very long to download, for example, some of these huge plink files, we're going to give you a memory stick. We'll pass it around. You can put it directly onto your hard drive so that you can download it faster. It takes a lot of time for some of these huge files. So we're also going to put up some exercises. I've already put up a couple for the first two sessions for this afternoon that we're going to be trying. Particularly, also going to include some solutions as well as exercises, and as well as I mentioned, there are links to software packages available on the website as well. So the website will be updated continuously, so that's going to be the main way for us to download data. Okay. So with that, I want to let Mike introduce himself.
So that's me. In case you could notice, there's a much skinnier, younger version of me, but not me nonetheless. So I'm an assistant member at the Fred Hutchinson Cancer Research Center. You can think of that as a slightly more communist version of an assistant professor. But I'm also an affiliate assistant professor here at UW. Prior to this, I was on the faculty at the University of North Carolina in Chapel Hill for several years before moving over to this side of the world. So my primary research is in high-dimensional data analysis, and that does include analyzing a wide range of different types of omics studies. So that includes everything from genomics, metagenomics, all the way down to proteomics. But recently, I've done a lot of work on DNA sequence analysis, and in particular, analysis of rare genetic variation, which is one of the key areas that we're going to be covering today. And I do some other stuff which is listed there, but that's not entirely relevant to what I'll be teaching here.
And so Mike's being a little bit modest, so he's also developed the SCAT method, which is the most used method for analyzing sequencing data.
And yes, it's named after poop.
So as for myself, so I'm an associate professor in the Department of Biostatistics here at the University of Washington. I'm also an affiliate investigator at the Fred Hutchinson Cancer Research Center. So you and I are just swapping, right? I'm affiliated one and then primary appointment at UW. So a lot of
us can spell biostatistics.
Yeah, all right, exactly. Yeah, oh. I didn't know that. Sorry, you should have corrected that.
I thought it was funny.
You let it happen. Thanks. So my research largely focused on genetic association studies. So a lot of the work that I focus on is how to account for correlated genetic data in genome-wide association studies. When I say correlated data, I'm talking about sample relatedness or population structure attributes, so how to account for that. And also how to take advantage of that to particularly potentially improve associated signals. So some of the work that I've been focusing on is how to infer genetic attributes from genetic data. So have a few papers on that. Also on how to estimate relatedness among individuals. So a lot of the methods that are out there assume that you either know the pedigree structure, so that if you're doing family studies, you know the pedigree structure, or you assume that sample data are unrelated. And so a lot of pre-processing and quality control is done to make certain that the individuals in your sample are related the way you think they are. So there's often issues with cryptic relatedness among individuals, including large-scale genomic studies where you may not have genealogical records. And so a lot of the work that I've been focusing on is how to infer relatedness among individuals using genetic data when genealogy is not available. And recently I actually started doing some work in pharmacogenomics with some collaborators trying to look at different responses to different drugs for different genetic profiles. So that's us. We would like to go around the room, briefly describe who you are, where you're from, what your research interests are. This was a pretty big class, so I guess don't tell us too much about yourself, but tell us enough so that we know a little bit about you. In the front.
My name is University of I'm a third-year student. I was enrolled in biochemistry department, but coming to the U.S. 2 years back, all what I've been doing is population genetics. So GWAS will be part of my work. I'm working on founder population, that is finding obesity, rare genes in obesity and diabetes in founder population. So it's hoping that this goes with that.
Nice.
I'm Holly Corbett. I'm at OHSU down in Portland. I'm going into my third year, and I'm trying to identify rare variants with association between ADSD and Down syndrome.
I'm Wes Bashir from Texas A&M University. I'm a second-year student there, and I'm studying hybrid sterility using cats as analysis.
I am Virginia Stanislas. I'm first year therapeutics to Matthew France. In Paris, I'm working on the deficiency syndrome.
And I'm Amy Flip from Paris as well. And I'm a biostatistician working on analyzing sleep data and sequencing data.
What disease? Are you saying particular traits or just general?
I'm Jose Miguel Uribe. I'm from the University of Costa Rica, the Department of Human Genetics, where I work with obesity and type 2 diabetes.
Hi, I'm Ian Bassini. I'm from Brazil, but I live in Chile. I am a PhD student, and I work with genetic improvement in salmon and Pilates.
Brian Green. I work at a small college in Salt Lake City, Utah called Westminster College. So I mostly teach, but I'm interested in neuroscience genetics.
Hi, I'm Monica. I'm a first-year student of Pearson Uni, UK. My project is on hip osteoarthritis.
I'm Diana. I'm studying PhD and I'm interested in sclerosis and thinking more about GWAS and
My name is Yemi. I have a MPH in biostatistics and epidemiology from UNC Fort Worth. I'm just interested in adding to my knowledge base and hopefully get a position where I can get to analyze genetic data.
I'm Crystal Grant. I'm a grad student at Emory. I'm studying DNA methylation.
Hi, I'm Simon Brother. I'm from Allelic, and I'm doing a master's in computational and system biology. And my project is related to concentration of cancer pharmacogenomics in sequence data.
I'm Brian Lohman. I'm at University of Texas at Austin, and I work on statistical epidemiology and gene study.
I'm from Brazil, and I'm doing my PhD in second year, and I work also in genetic improvement in statistical, trying to identify the genetic variant and associate it to resistance.
My name is Rob Baker. I'm a postdoc at the University of Wyoming, and I'm interested in microarray
is John. I just graduated from my bachelor's like a month ago. And
Where? Here?
Where?
From where?
Cal Poly.
Okay.
In California. So I'll be a first year PhD student at UCLA.
What department?
And,
What department?
Biostats. And I just thought statistical genetics was interesting, but like at my undergrad we don't teach anything like it, so it's good to be finally like getting some knowledge about it. So it's like a crash course.
Okay.
I'm Barb Bakery. I was a postdoc in biology graded up, and I recently accepted a job at Fred Hutch as a statistician, and so I'm catching up to speed with some of the things I've been doing.
I'm Sarah Bartley. I'm a PhD student in genetics in Calgary, and I work on therapy and statistical reason exome sequencing.
I'm Kate Shura. I'm a postdoc at NIH, and I'm working on an exome sequencing project on rare human diseases. My name is Alba Strand. I'm a third year graduate student at Columbia, and I'm looking at patterns of recombination and mutation rates in humans.
Hi, I'm Cindy Polito. I'm a third year student at Wayne State University, and I'm looking into regulatory variation.
I'm Whitney Dolan. I'm a graduate student at Purdue University, and I study transcription regulation of metabolism in rhabdomyocytes.
I'm Maddie, PhD student from the University of Queensland in Australia. I'm studying patterns of recombination during speciation in plants, so sequencing data in genomes.
Hi, I graduated with an undergrad degree a couple of years ago and I'm working on tech. And I'm using since at the Moore Lab of Zoology at Occidental College in LA. And I'm using sense and sequencing data to study progression of convergence across the hybrid birds.
My name is Cameron. I am a sixth-year PhD student at West Virginia University. I'm trained as a clinical psychologist, and my work is in the interaction of fear, anxiety, and pain. And I'm doing some work in genetic predictors of pain sensitivity, and how those predict things like fear, painful medical procedures, and fear.
I'm Sophie Mayer. I'm a first year graduate student in epidemiology here at UW. I'm a research assistant at Armitage. And I have interest in pharmacogenetics.
My name is Carlos Santos. I'm from Brazil. I work for Embrapa. I'm a group leader. And I have a PhD from UW Medicine, 15 years ago.
Welcome home.
I'm Java. I'm a physician from Brigham and Women's Hospital. I work on genetics and chronic obstructive pulmonary disease.
I'm Lucia. I'm a grad student at UCLA, and I'm studying
Hi, I'm Lori Norberg. I'm an assistant professor at the University of Kentucky College of Dentistry, and I study communication.
Maybelline Gamboa. I'm a third year PhD student at Colorado State Smithsonian
it's a diverse group. So heard a lot of doing GWAS and sequencing study, which is perfect. So the way we're going to divide the lectures is that we're largely going to focus mostly on GWAS for the first part of the lecture, first part of the module, and then the second part, second half, we're largely going to be focused on sequencing studies. Okay. So if there aren't any questions, let's get right to it and go to the first lecture, which is on case control association testing. So, presumably everyone's familiar with association mapping. It's now widely used to identify genetic variants that are associated with complex traits. So with the advancement of technology, it's now common for genetic studies to have data on 100,000, even millions of genetic markers across the genome. And so there are different ways to kind of analyze and identify which variants may be associated with the trait of interest. But the most simple approach is to test each single marker individually and to see if it's associated with the disease or a binary trait in samples with unrelated individuals. So let's first consider the case where we're interested in a dichotomous trait. Either you have the trait or you don't, you have a disease or you don't. And so essentially, at its most basic, case control association testing is trying to test for independence between trait and allele or genotype. So if you tell me your allelic or genotypic value at a particular marker, does that give me any predictive power for determining whether or not you have the disease or not? Okay, and so we want to know whether or not those two are independent. So when you're doing case control association testing, there are essentially two ways to do it. So one, there are two classes of tests, I would say. So one class are the allelic association test. Okay, and so the allelic association test, the allele is treated as a sampling unit. Okay, so typically you're making an assumption of Hardy-Weinberg equilibrium, that the two alleles within an individual at a marker are independent. Okay, so if you have in individuals, then essentially your sample size is 2n, all right, because you're assuming that the alleles within an individual are independent. So alleles within an individual give, so conditional on trait value, the two alleles are conditionally independent. Okay? So genotypic association test, the individual sampling unit. So it does not necessarily assume Hardy-Weinberg equilibrium. So if Hardy-Weinberg equilibrium does not hold, if you don't have independence between the markers, genotypic association tests are still valid. Association tests in that setting. So those are the two classes of association tests if you're testing a single marker. Allelic test where you assume that the alleles are independent from the individual, conditional on trait value, and the other one is genotypic where the individual. So you essentially model whether or not you have genotype big A, or big A, little a, or little a. Okay. So for allelic association testing, the Pearson chi-square test was one of the first tests used for that. So this test, hopefully people have heard of it before, essentially looks for deviations from independence between trait and allele. So let's say we're at a SNP and we're interested in knowing whether or not this SNP is associated with case control status. And so we can arbitrarily label the alleles as 1 and 2. You may label as big A, little a. It doesn't matter. We're just labeling the alleles, and there are two different types. Okay. And so we're going to let in our sample N subscript CA be the number of cases, N subscript CO be the number of controls who have genotype data at a marker. So with the Pearson chi-square test, you can essentially write the data in a contingency table, okay. Whereas you can look at the number of cases that have allele type 1, okay, so that's the number of cases this number of controls that have allele type 1, number of cases that have allele type 2, and the number of controls with allele type 2. Okay, so we have our data, we can write the data into a contingency table based upon the number of cases and controls that have the different allele types. Okay. Whereas for the Allelic test, we treat the allele as a sampling unit. So individuals who, for example, cases, okay, number of type 1 alleles in the cases is 2 times the number of homozygous, 1-1, right? Because if an individual is homozygous 1-1, they have 2 alleles of type 1, and they get counted twice because we're treating the allele as a sampling unit, okay? And similarly, the number of heterozygous can contribute one unit to this cell. Okay? So N1 is the number of allele type 1 in the cases. N1CO is the number of allele type 1 in the controls. N2 is the number of allele type 2 in the cases. N2CO is the number of allele type 2 in the controls. Okay. And similarly for the lower right-hand corner, okay, this is number of type 2 alleles and controls. Individuals who are controls who are homozygous for the type 2 allele, I mean, they contribute 2 alleles to that cell. Individuals who are heterozygous in our controls contribute one unit to that cell. So the null hypothesis in this setting is that the alleles, allelic type, and case control status are independent. So the null hypothesis is there is no association between the row variable and the column variable. Okay? So if you tell me that individual is a case or control, that doesn't tell me anything about what type of allele they have. Okay? And similarly, if you tell me that an individual has allele type 1, that doesn't give me any information as far as the individual's case control status. So that is the null hypothesis. There is no relationship. There is no association between the row variable, allelic type, and case control status. The alternative is there is an association between the 2 variables. Okay. So you can use a Pearson chi-square test for independence for this. Okay. So essentially observed cell minus expected cell count over expected cell. So understand that the expected cell count is what we expect under the null hypothesis. If there is no association between the trait value and the allelic type, then what would we expect the cell number to be? So the expected cell count is row total times column total count, but that's not really intuitive. Why is this the expected cell count? Okay. And we'll talk about that in the next slide. But under the null hypothesis, then this test should follow an approximate chi-square distribution with 1 degree of freedom. Okay. So, one question: why is it a 1 degree of freedom test?
Because you have 2 things, so N minus 1.
Yeah, but okay, so what does that mean, degree of freedom, though? So essentially we're saying that the margins are fixed. So that means that if I tell you one value here, I can fill in the other 3 values, right? Because the number of allele 1s, we know that. The number of allele 2s, we know that. We know the cases, we know the number of controls. So if I tell you one of these values, then I can fill in the other 3. We can fill in the entire table. So that's 1 degree of freedom. Because if you know one value, then that gives you the other 3 cells. Okay? So there's 1 degree of freedom here. So let's think of this notion of no association a little more. So what do we mean by no association? Okay, so first, let's think of a real example. Okay, so this is coming from a paper that was published in 2010 where they were looking at for an association between some candidate SNPs and a phenotype that's responsible for blindness and has a high prevalence in Thailand. So in this study, there were those who had the CC genotype. There were 6 cases, 10 controls. For heterozygous, 8 cases, 6 controls. And homozygous for the T allele, there were 75 cases, 163 controls. Okay? So this is the data that we have, so we can turn into a 2x2 contingency table easily. right? So does everyone know how we get from here to here? So for the cases, okay, how do we get allele C? How do we get this down?
2
times the 6 plus the 8.
Yes, 2 times 6 plus 8. So those who are case who are homozygous for C allele get 2 times 2 units contributed. That's 12. And those who are heterozygous give 8. So 12 plus 8 is 20. Okay? So we went from genotypes to allelic types, and now we're going to do a chi-square test for independence. We want to know whether or not there's independence between allelic type and case control status. Okay. So the intuition for this test is, let's suppose the null hypothesis is true, that there is no association between allelic type and case control status. What counts would we expect? So let's think about this for a minute. Let n be the total number of alleles in the sample. So if we assume independence, the expected number of case alleles that are of type T is. So we want to know what are the expected number of cases that are type T, what we expect. So if the sample is n, probably allele is from the case and allele is from control. What is this assumption making right here? How do I go from this step to this step? They're independent. So probably allele is from a case and allele type is T. It's probably allele is a case times a probability allele is a type T. We're assuming independence here. So that's the null hypothesis to go from here to here. What's probably allele is from a case? How many case alleles are there? Okay, allele is from a case. So essentially, 178 of the alleles are from cases. What's probably alleles of type T? 500 over 656. So the expected number of cases, allele type T that's from the cases of 149. Right. So does everyone see that? So that's the Pearson chi-square test. We're assuming independence. So under the null hypothesis, we would expect 149. Okay. What did we observe? We observed 158. So you would do the same calculations for the other cells as well. And so if you fill in the table, you get this. And so essentially what we want to know whether or not this table that we have of expected counts, how different is it from what we observe? So how different is this observed data from what we expect if indeed there's independence between allelic type and case control status? And so the chi-square test does that. So it's the observed value minus expected value squared divided by the expected. And so this chi-square statistic has a value of 4.369. The p-value, what's the probability of observing a statistic at least as large as what we observe? And so you can use your favorite software, and for chi-square 1, the area to the right of that, and it'll be 3.7%. Okay, so the p-value is 0.0337. So any questions about that? So that's the basic idea behind the Pearson chi-square test for genetic association with alleles. Okay. In some settings, you may have a situation where the number of samples, number of alleles, okay, can be less than 5. So in this example here, this distribution, this chi-distribution assumes that you have enough samples such that normality holds, and chi-square is just a normal distribution squared. Okay? So we have to have enough samples such that the asymptotics hold. So we assume some normality of the test statistic squared. Okay? So in many cases, the assumption of normality is not going to hold, particularly in the small sample setting. Okay? So a lot of times you may have sample sizes that are quite small, and then assuming a chi-square distribution is not valid. Okay? And so in those cases, you may want to do an exact test. Okay? So the rule of thumb typically is if you have a setting where you have a condition table, for example, where one of the cells is less than 5, okay, or an expectation, okay, then you probably don't want to use, for example, a chi-square test, a Pearson chi-square test, right? Because the assumption of sample size is large enough such that normality holds what might not be valid. And so the Fisher-Zach test can be used in those settings. So in this case, you have, for example, a much smaller sample. Okay? And so we have 24 cases. So in this case, we have 24 case alleles, 24 control alleles as well. So much smaller set. So we want to know whether or not there's an association between our trait value and case control status. And once again, the chi-square test is not going to be appropriate for this data set because sample sizes are too small. So in this setting, you can actually use an exact test. And so similar to what we did before, you really only need one of the random variables to do the test on, because once you know one random variable, you can fill in the remaining cells. Okay? So I'm going to use X to be what I'm going to base my test on. So let's X be the number of case alleles that are of type T. So we're going to base a test based upon the number of case alleles that are of type T. So we observed 21. So once we know this is 21, since the margin was fixed, number of cases is fixed, number of controls are fixed, if this is 21, then we know for certain that this has to be 3. If we know this is 3, we can fill in the remaining cell. So we're going to base a test this based upon the number of case alleles of type T, but you can totally base a test this on any one of these cells. You can base on the number of control alleles that are type T. So under the null hypothesis, and you can look up the Fisher-Zach test, the distribution of the number of alleles that are type T, okay, is going to follow a hypergeometric distribution, okay, where the probability under the null, if there's completely random shuffling of the alleles in case control status, under that setting, this will be the distribution, where probably that X is equal to some value little x. Is 35 choose X times 13 choose 24 minus X over 48 to 24. So quick question, what are the possible values that X can take? Any thoughts? Can X take a value of 10? So if this is 10, what must this be? 25. But we don't have 25 controls. Okay? So there's a range of values based on the data that X can take. Okay? And this is the distribution. Okay? So the range of values that X can take is from 11 to 24. So those are the possible values that it takes. So what's the probability if we randomly shuffle the alleles and gave them to case control? What's the probability that 11 of the alleles are going to be case alleles of type T? Okay? So X, once again, is the case alleles that are of type T. What's probably that X is equal to 11? It's just 35 to 11, 13 to 13, and 48. So 0.0001. So 1 times e to the minus 5. Very low probability. Question?
You said that there is a 24 possibility for the X case of type T to be 24.
So X can take on values between 11 and 24.
Why not 21 if I have any in the table, a case for any case 21?
So that's what I said. So X can take values from 11 all the way up to 24. We observe X equal to 21.
Also, so that's an observation.
That's what we observe. Now we need to know how likely is it that we would observe this data under the null if indeed there's no relationship. So we're getting all the probabilities under the null hypothesis for X equal to 11, 12, 13, all the way up to 24. So we need to know how likely is it for us to observe the data that we observe. if indeed there's no relationship between trait value, case control status, and allelic type. So any questions about that? All right?
Is that 35 divided by 11, or is that the range?
So yeah, so this is 35 choose 11. So this is combinatoric. So the question is, so 35 choose 11. Sorry if you haven't seen me choose from. So okay. So this is like how many different combinations if there are 35 different ways. If I wanted, let's say I have 35 different color balls, how many different combinations of 11 can I get, right? So it's 35 choose 35 factorial 11 factorial over I think 24 factorial. Yeah. So the formula is n choose x, for example, is n factorial over x factorial over n minus x factorial. Okay. Where n factorial, for example, is n times n minus 1 times n minus 2 times whatever, times all the way down to 1. Okay. So it's how many different ways can you choose different subsets. So if you do this calculation, this is what you would get. So what's the probability that x is equal to 11? It wants to be to the minus 5. 12.03, 13.04. So we observed x is equal to 21. So we had 21 case alleles of type T. If we wanted to construct a two-sided, what's the likelihood of observing something at least as extreme as 21? Okay. Any thoughts on that? So we can construct a test. So we observe, so our test statistic is we observe x is equal to 21. So we want to know what's the probability of observing something at least as extreme as 21. So that's why the answer is 1, but it's a two-sided test, right? Because you could be on the negative side too, on the other side, or plus probability x less than or equal to, and what's the one? 14. I saw a quite hand in the back.
I see. So you can get v1 minus the probability that x is less than 20.
Exactly. Yeah. But, okay, so you got to think about this a little bit more. Are you doing a two-sided test or a one-sided test? If it's a one-sided test, you want something at least as large as 21, then it's a one-sided test. Then you can add it up. But the two-sided test is, okay, you don't know whether or not you're going to get something larger than what you say, or smaller, right? So the critical value could be 21, greater than 21, or less than 14.
You subtract out the...
Or you can just add it, right? What's probably of finding something at least as large as 21? 0.021 plus 0.04 plus 0.03 plus 0.001.
Right.
Plus, if you're doing a two-sided test,
right.
What's the opposite end? So for example, if you're typically doing a two-sided test for a continuous distribution, and let's say your critical value, your data is right here, you typically find the opposite end too for a two-sided test, right? You want something at least extreme. That's what you observe. This is the same principle holds here, except now we're talking about discrete values. So what's at least as extreme as 21, okay, on the lower end, it's 14, 13, 12, and 11. Question?
So you're going into the probabilities here, but the big picture of things, right? So we do the Pearson S-squared test. You did it earlier.
Yes.
And you had a probability of 0.03.
Yes.
Okay. Tell us what that means in the big picture.
So in the big picture thing, that means the probability of finding something, the association, as least as large as the number that we observed.
Right.
Okay. If there's no association, only 3.7% of the time will you find something at least as large. Okay. But once again, this is something squared. So this actually covers this case, right? Because the chi-squared, this is, for example, a normal distribution. Chi-squared is the normal distribution squared. So these values get squared, and so all the values are positive. So this chi-squared is automatically a two-sided test.
Right. So under the conditions of 0.03,
Yes.
do we have an association?
What's your significance level?
0.05.
If that's your significance level, then yes. So that's the question. So in this case, we're constructing
If it's significant, then we have a significant association.
at the 0.05 level.
Right.
That's right. It depends on what level you're at. If you're talking about genome-wide significance, then no. If you're talking about like humans, like that's 5 times 10 to the minus 8.
Right. So under this condition here, at 21, we're at 0.02, and you were at 0.05 level.
You're not at 0.02. That's the question. This is what you observe for x equals 21. You need to find something at least as extreme as this. So we look all the way, so we want something in the case for chi-squared distribution, we looked at everything to the right of this value. So we're doing the same thing here, but we had to construct a two-sided test. And so to construct a two-sided test, it would be the following. So rejection region would be x equals 11, 12, or 13, 14, 12, 22, 23, 24, right? Because 14, 13, 12, 11, this gives us 0.025. This gives us 0.025. So this test is 0.025 plus 0.025, 0.05. Okay? So at the 0.05 level, since x is in our rejection region, what do we observe at x equals 21? At the 0.05 level, will we accept or reject the null hypothesis?
I don't get why are we doing from 21, 24, and 14 to 11?
Because the null hypothesis, what's the probability of finding something at least as large as what you observed? Okay, so that's how you do the hypothesis testing. What's the likelihood of finding something at least as large as what we observed? Okay, so that's our rejection region. So here, for example, when you're doing, for example, a normal distribution, your z-score, your significance level is based upon the probability of finding something at least as large as your value that you observed, and that's the area to the right and to the left. So you don't just do it by where you're at; it's something at least as large. Okay? So that's just basic statistics, for hypothesis testing. So when you determine whether or not the value that you observe is significant or not, okay, you look for values that are at least as large as what you will have observed, and that is your that's your rejection region. What's the probability of finding something at least in this region, and that's your significance level. But once again, if you have small data sets, okay, or small counts, okay, where chi-squared normality may not hold, then you have to do some type of exact test. Okay, that's one approach. There are other approaches as well. Okay, but this is the Fisher exact test. Okay. So at the 0.05 level, back to my question, I reject if I see values, if my value is 11, 12, 13, 14, or 12, 21, 22, 23, 24. My value was 23, so I would reject the null hypothesis. So the nice thing about exact tests is that it allows you to easily construct your rejection region. Okay, so you can easily do one-sided tests as well. The drawback about it is that it's not smooth. It's not a smooth rejection region. Okay? It jumps. For example, if you were to go from the rejection region, for example, 22, 23, 24. For example, there is no 0.025. There is no, for example, 0.01 test. Okay, so if you wanted to construct a test that was level 0.01, you can't do it so easy. Whereas in the continued case, you could. Question?
So when you say that you would reject, are you saying you're rejecting the null hypothesis?
I reject the null hypothesis. There is significant evidence that there is a relationship between your allelic type and case control status. Okay. Because an X is equal to 21 is extreme. Okay. If X was 20, would you accept or reject? You accept or not. Or the proper term is you can't reject. You have not enough evidence to reject. Okay. Because you have p-value of 0.072 greater than 0.072. Questions?
So your upper and lower boundaries are always determined by your experimental observations?
Yes, that's right. Your question is, so your p-value is based on your data, your observed data. What's the probability of observing something as large as what I see in my data, if indeed there was nothing going on? So the no just means there's nothing going on. How likely is it for me to observe a value that I observed in my data, if indeed there was nothing going on? So p-value is data specific. It depends on your data.
So it doesn't depend on confidence interval, because I got an impression when you chose 21, 22, 23, the corresponding p-value was 0.025,
Yes.
which is the half of my, for two takes, it's half on the right and half on the left, according to 0.05 confidence that I chose. So I
asked you to construct a 0.05 confidence interval.
So that's a constructed one. It's
Yeah. So regardless of what you would do that even before seeing your data. What is before I'm telling you got a 21, you construct what's going to be accepted, what's not. And a 0.05 confidence interval is X values 21 and higher and 14 and lower, we reject.
For a two-take test.
Yes. And if you want to do a one-tail test, let's say you have some prior belief, in which in most cases we know in genetics that basically I know there's going to be large values are going to be associated. If you have some prior belief of that, you can construct a one-sided test. If you did a one-sided test, then the alpha, if you excavate 21, the alpha level will be 0.0225. Okay. But most times, since you don't have a lot of information, you could do two-sided tests. But if you have some prior information that you want to test, okay, you know what, I want to look for values that are larger than what this is what you see, then you could do a one-sided test easily. Questions? Okay. So let's talk about the Armitage trend test. So this is the most common genotypic test for unrelated samples today. So essentially, it's essentially doing the same thing. You're testing for association between a single marker and a trait. Okay. But now, for example, you let Y is equal to individual I is homozygous, 1 if I is heterozygous, and 0 if homozygous. Okay. And we let X is equal to case. X is 1 if it's a case, and 0 if it's a control. So what you could do is just do a simple linear regression model, okay, on genotype onto alpha. Anything strange about this model? So Y is my genotype and X is my phenotype. They're both discrete. They're both discrete. Okay. More than that, right? I'm treating the outcome as my genotype and my case control status as my predictor. So I'm trying to predict allelic type or genotype value based upon case control status. All right. So in the linear regression model, what is the assumption? Typically, an assumption is that what's random in the linear regression model? The response. Why? The response is random. Okay. The predictors are fixed. If you think of a case control design, is case control status really random? Because you sample based upon case control. You know who's a case, you know who's a control. Right? So you're sampling people based on, oh, you're a case, I want you in my study. Okay, I need more cases. I need more control. So there's no randomness there. What is random? Genotype. Genotype is always random. You never know a priori a person's genotype. If you're doing a case control design study, you know a priori who's a case, who's a control. But it turns out that it really doesn't matter because at the end of the day, we're just looking for an association. So whether or not I put X on that, I put case control status as my response and genotype as the predictor, the association doesn't care about that. So if I'm testing whether or not there's an association, it doesn't matter what's going to be the response, what's going to be the predictor, because we're just looking for linear relationship or association. Okay? Just like here, it doesn't matter if I put allele on one axis, I'm going to let alleles be the rows and the case control status be the columns. It doesn't matter, because we're just looking for association. Is there a relationship? Question?
On sample size, how do we know that the sample size is too small to validate?
So that goes back to what I said before. Rule of thumb, typically, if you have a cell that has less than 5, then you better be worried about asymptotic assumptions like normality or chi-square distribution holding. Okay? So if you have at least one cell that is less than 5, and here we had 3 here, then you need to be concerned whether or not the assumptions of normality or chi-squared or whatever asymptotic assumptions that are being made are appropriate. Okay? Okay. So essentially what we're doing is, so if there's no association between allele type and case control status, then what is beta 1? 0, right? Beta 1 is 0. This gives us no information. It's not a good predictor of y. It doesn't tell us anything about it. And even if you do it the other way, it's still the same. Is beta 1 different from 0? Is there an association? Okay. So we're testing beta 1 is equal to 0. You can do a linear regression. But you can actually show, and this is why it doesn't matter what is your response and what is your predictor, because you can show that this test, since beta 1 hat squared over variance beta 1, is actually equal to your sample size times the correlation squared of x and y. Correlation doesn't depend on what's the response and what's the predictor. So that's why it doesn't matter what is the x and what is the y here, right? Because correlation was correlation between height and weight. Same as correlation between weight and height. Doesn't matter. Okay? So that's why it doesn't matter if we're just testing for association. Is there a linear relationship between the two? Okay. So note that the variance estimate is basically sum of square deviation of y. It's not the fitted values. There are little technical, there's different ways how you can estimate the variance. But this is essentially the Armitage trend test where you're testing for association. But the nice thing about it is that it doesn't break the alleles into 2 separate groups. So for example, if you're homozygous, then you get a value of 2, 1, and 0. But it doesn't assume that you can automatically break the alleles, treat the alleles as sampling unit. Each individual has their own value. Okay. So if you wanted to do the same data set and apply the Armitage trend test, we get a p-value of 0.0553. Okay? So this doesn't assume independence of alleles, doesn't assume Hardy-Weinberg, p-value 0.053. Is that significant at the 0.05 level? Guess it depends if you want to get published. Because many people say, you know what, I'm going to round down. Okay? So these are things to think about. Okay? Is this significant? Question?
So if I have a situation where homozygotes for one allele are incredibly rare, and so I'm lumping, for example, anyone who has 1 or 2 C's into one category, and then T homozygote is the other category.
Mm-hmm.
Which way, what is that, right? I'm not counting alleles, I'm using genotypes.
Yeah.
But I've pooled these two.
Yeah.
Acceptable if you use this test?
Yeah, but so it's fine, and the reason why it's fine in this setting is because we're not treating slideshow. Sorry, it's fine in this setting because we're actually treating y kind of like a continuous variable. We're not putting them in groups. If you put them in groups, then you run into problems. For example, if one group, that goes back to here, if you have one group that has a low sample size, for example, like 3, you have a cell. Really here, there are no real cells, right? Because we're just saying, we're treating Y kind of like a continuous variable, right? So even if you don't have any at all in a certain group, I mean, then it doesn't matter, because then it's just like 0, 1, which is perfectly fine. Okay? Because we're treating it as continuous in some sense. Question?
Can you just comment on when you use this or why you use this over chi-square?
So if you're concerned about the assumption of Hardy-Weinberg equilibrium holding, if you're assuming, so Hardy-Weinberg assumes that the alleles within the individual, like there is random mating and things of that nature, right? So this doesn't assume that, right? So if it's random mating, then if I know one of your alleles, that doesn't give me any information about another allele. But if you have assorted mating, for example, let's say, for example, one individual who is AA is more likely to mate with somebody who also has big A, then the other method, assuming independence, doesn't hold. But in practice, it's not going to matter. And the reason why it's not going to matter is that most of the time when you're doing any filtering, you're filtering based upon Hardy-Weinberg equilibrium. So you're tossing any SNPs that deviate from Hardy-Weinberg because you don't trust those, right? Because there has to be some plausible explanation for why you have like large deviations from Hardy-Weinberg equilibrium. So in practice, it doesn't really matter because you're going to do quality control filtering based upon Hardy-Weinberg equilibrium, and these two tests are actually equivalent if the data are in Hardy-Weinberg. They're not equivalent if there's Hardy-Weinberg disequilibrium. Okay. Question?
When you're looking at case control data, is it proper to do Hardy-Weinberg on only the controls, or should you always do it on a mixture of case plus?
Yeah, it's hard to say that. And the reason I say that, if you're at the marker that's causing the disease, then you don't expect it to be in Hardy-Weinberg. Okay, and so there was a nice paper by Nancy Cox when she was at Chicago, and I forgot, it was in American Journal, I'll point it to you, where they have a paper on like rational inference on deviation from Hardy-Weinberg equilibrium. So in some cases, you expect, particularly at the causal variance, you should have some deviation. And there's some methods out there that actually do gene mapping based on Hardy-Weinberg equilibrium. They go through the genome and say, oh, any markers that have deviation from Hardy-Weinberg, that's a candidate for disease. Right? And so that's kind of, so there's some work on like Hardy-Weinberg disequilibrium mapping to look for those regions of the genome where there is deviation from Hardy-Weinberg, and maybe that tells us some information as far as this region being associated with the disease.
What was her last name?
Cox, Anna, huh?
Nancy Cox.
Yeah, Nancy Cox. I think she's a senior author. Anna Pollution, I can't spell that. But yeah, just American Journal. I have it somewhere. Just remind me. Okay. So another way to do the analysis when you're looking at genotypes are odds ratio. This is actually the most popular way to do case control association studies with binary data, is to do logistic regression. Okay? And so we're going to talk about that. So let's first talk about what are odds. Okay? So in this case, for example, in our sample, those individuals who have the big A genotype, 66 survive, 6% survive, 33% die. Those who are heterozygous, half of them died and half of them still survived. So you can think of some survival time. So you look at some patients over time to see, like, what's their chances of survival over time after, for example, some surgery, for example, an invasive surgery. And for example, those who are homozygous AAs, 33% died. Those who are heterozygous 50% died. And those who are homozygous with little a, 90% of them died. Okay. So first question are what are the odds? So what's the odds of this person? If you have the big A genotype, what's the odds of this individual surviving after 10 years? 2 to 1. Okay. 33%. This is the probability of death. Okay? But the odds are 3 to 1 or half. Okay? Similarly, what's the odds of death after 10 years, probably survival? What's the odds if you have a little a genotype? What's the odds? It's what? 9 to 1. That's your odds. Okay. So the question now is, what's the odds ratio of those who have the little a genotype relative to the big A? What's the odds ratio?
You divide 1 over 2 over 9.
So this is the reference, so 9 over 1 divided 1 over 2, 18. Okay. So you're 18 times more likely, odds 18 more likely to get the disease, to die, for example, if you have a little a compared to the big A genotype. Okay. So that's the odds ratio. All right. So the odds of disease, odds of death, are 9 over 9 to 1. Okay. So you're more likely to survive here. If you're heterozygous, it's even money. You may 50-50 chance of survival. But if you are homozygous for little a allele, it's not looking good. Okay. Okay. So when you're looking at odds ratios, okay, you typically choose some reference genotype. Okay, and what are the odds of disease relative to some baseline genotype? So in the previous example, I said, what are the odds of disease for the little a genotype relative to the big A genotype? Odds ratio is 18. All right. So typically you're going to choose some type of reference genotype. Okay. And in this case, I'm going to let the CC genotype, for example, be the reference genotype. So the odds ratio of the TT allele, so in this case, I have big T, heterozygous, and little c. And I'm letting A be the number of cases that are type big T. Number B is number of controls, A prime, C, D. So everything's going to be relative to baseline. So odds ratio of the homozygous T genotype is the odds of disease in an individual with the TT genotype relative to the odds of disease in an individual with the CC genotype. Odds ratio of CT are the odds of disease in an individual with the CT genotype relative to the odds of disease for individuals who are homozygous for the C allele or the CC genotype. So what does the odds ratio of 1 mean?
Reference over reference.
Not reference over reference. Reference is in the denominator. So I'm going to interpret. There's no the odds are the same. Whether or not you have the big T allele or the little c, your odds are the same. So there's no association or there's no difference between your risk of disease if you have the big T genotype relative to the reference. So odds ratio equal to 1 implies there's no association with the disease for the big T genotype relative to the CC genotype. Okay. And similar for the odds ratio for the heterozygous. If you have an odds ratio that's greater than 1 or less than 1, that applies an association with this disease, the true odds ratio. But this is statistics, right? So you're going to get some odds ratios that are going to be greater than 1. Are they significantly greater than 1? Okay. Or is this due to random noise? Okay. So we need to have some type of standard errors for our estimates. Okay. So logistic regression, has everyone done logistic regression, or have you heard of logistic regression? All right. Question?
Just in the last one.
Oh, okay. All right. So, all right. We'll cover it here too. Okay. So logistic regression is used to get odds ratio and confidence intervals. Okay. So we're going to let pi I be the probability that individual I is affected with the disease, and let GI be the genotype of individual I at the SNP. Okay. So PI is the probability that you have disease. What is 1 minus PI? You don't have the disease. So what is PI over 1 minus PI? That's the odds. Right? That's the odds. Probably you get the disease divided by probably you don't get the disease. Okay. That's the odds. Okay. That's the odds of disease. So log of pi over 1 minus pi. So given that you have genotype, given the person has a certain genotype, what's the log odds of disease? So pi 1 minus pi I is the odds, and we're taking the log of it. So it's called the log odds. So what are we going to have to do, for example, to get the odds? If we have the log odds, what do we need to do? Exponential, right? To get the odds. Right? So remember that. Okay. So the log odds of disease, for logistic regression, we model log odds of disease, pi 1 minus pi, is equal to beta naught plus beta CT indicator that the genotype is CT, so beta TT indicator TT. So let's write it down. So this means that if a person has the CC genotype, what are we saying? We're saying that the log for person I, the log odds, given this individual has the CC genotype, what are we saying his value is? Is that for beta naught? And let's just assume that we're in a setting where it's a process study. So it's beta naught, for example, right? What's the log odds if the person is heterozygous? Can someone tell me what that is? According to this model. So this is an indicator. This is 1 if the person has genotype CT, 0 otherwise. So the person has the CT genotype, what is?
Beta CT.
Beta naught plus beta CT. And similarly, what is it for a person who has the TT genotype? Plus the other one. Yeah. So that's our model. Okay. So just looking at this, if we're comparing these groups, if there's no association, what do we expect the beta CT and beta TT to be? 0, right? Because everything's going to be relative to CC. So all these beta CT, beta TT should be 0 if there's no association. Okay? So how do we get the odds ratio? So what's the odds of disease, for example, if you have the CC genotype? What's the odds of disease? How do I get my odds? E to the beta naught. That's the odds of disease, for example, if you have the CC genotype, right? What's the odds of disease if you have the CT genotype? Beta naught plus beta CT. What's the odds of disease if you have the TT genotype? Plus. So what's my odds ratio of if CC genotype? Give me the odds ratio. What's the odds ratio of the CT allele? Everything's relative to baseline CC. What's the odds ratio? So what's on the denominator? E to the beta naught. What's on the numerator? What is this? E to the beta naught, E to the CT, over E to the beta naught. What are we left with? Beta naughts cancel, and we're left with E to the beta CT. So this is an estimate of the odds ratio that we can get from our model. Any questions about that? So that's how we were doing logistic regression. That's how they're getting the odds ratio. Question?
So we just arbitrarily chose CC as our reference?
That's yeah. So most of the time, the convention typically is to choose the major allele, the one that's most common as the reference, because presumably if it's a disease, then it's not going to be common disease where more people have it than not. So it's probably going to be a more rare allele. So but that's something to be thinking about. And one of the exercises, there's some things you have to think about with that as well. Okay. But typically, you're going to choose one of the homozygous ones to be the reference. All right. So we're going to, sorry, that's the wrong, I put the wrong address on here. So use the web address from the that I gave you. But we're going to use in our software package to analyze this data. We can also do odds ratio based upon allele counting as well. Note that if you're going to do this, you can't do this by hand. You really need a computer because things have to converge, and so you're not going to do this by hand if you're going to do genotypes. So you're going to use some software. If you're doing an allelic test, if you're okay with making the assumption that the alleles are within an individual independent and similar to what we did before for the Pearson chi-square test, we can do this similarly, but now the odds of disease actually has a nice form, and you can look at it a little closer a little later, where you can easily get the odds ratio, and it's nice. If NA is the number of cases that are type T, NB is the number of controls that are type T, NC is the number of cases that are type C, and ND is the number of controls, then it's nice with the odds ratio, for example, you're comparing, instead of comparing genotype based on genotype, you have to just choose the baseline allele, which allele. But it really doesn't matter if we just have two alleles. It doesn't matter which one's the baseline, which one's not, as far as an association. And the odds of disease, odds ratio is just N times D divided by NB times NC. It's actually a really nice formula. Question?
So along the same lines of Fisher, when you have like a small sample size and one of these cells is 0, you can't divide by 0, so can you not have an odds ratio?
No, you're not going to have it. No. Yeah.
There's
So
no other way
you so you wouldn't get an odds ratio because you can't divide by 0.
Yeah.
Anyways, yeah. So you can't do an odds ratio in that setting. So you might have to use some other approach. Yeah. If you have cells with 0. All right. Here, these are some of the formulas. You can easily do this by hand. If you just have a 2x2 table, you can easily get the odds ratio in that case using these formulas. But most times you're probably going to use like logistic regression or some software. But it's to show you that if you do have a 2x2 table, you could do this by hand as well. And then here's an example where I have the odds ratio of 1.7 for the Leber's hereditary optic neuropathy. And so, would you reject the null hypothesis?
Yeah.
Why? Now we got to talk. So this is our estimate, but there's some noise, there's standard error. So we got a confidence interval. This is the point for confidence interval. So the confidence interval, upper confidence interval is 2.92. The confidence interval for our odds ratio. So our odds ratio estimate is 7.3. Okay. Our standard error. So we have our standard error. So our confidence interval, the 95% confidence interval is equal to 1.03 to 2.92. Would we reject this is a 90%? Would we reject the null hypothesis? Okay. We reject the null, right? Because one isn't in a confidence interval. Odds ratio 1 is saying that there's no difference, right? So the odds are the same, whether or not you have the T allele or the C allele. 1 is not in our confidence interval, barely. Okay. And so since 1 isn't in a confidence interval, we can reject them on some level. So any questions about that? Okay, so now it's time. So we have about 10 more minutes. We'll go over the solutions quickly over the break, but the lab is here. So session 1. These are we have some exercises for you. Sorry, let me see. Let me not have some here. So so if you go to the course website, we have exercises for you to work on, and I'll also pass them out here in case people want to feel more comfortable looking at a sheet of paper. And then there's an R script that I've already written to help you use R to answer some of the questions as well. Okay, so for those who are not familiar with R. Right. Question.
The equation that they have just described, are those for only case control or if it was the same with related?
With who?
Related.
Related?
Yes.
No, they will not work. This is for unrelated. Yeah, so we'll talk about how to handle relatedness a little bit later, but what we did today was talking about unrelated individuals in case control design. So there's specialized techniques to analyze samples with related individuals. And the exercises are also online as well. Everyone get enough work? Yeah, and it won't include one. Yes. Zero.
Oh, okay. Yeah, you can get that when you check it.
Yeah. So there's a break at three over at the South Campus Center where they have lots of goodies and snacks and everything. So feel free to work through the break. We'll reconvene here at 3:30. But if you want to break and eat and come back, that's perfectly fine. We'll go over some of the solutions to the problems after the break. Okay, so feel free to step out and grab some goodies and come back.